Hi, welcome to the Bioinformatics Chat. Today I'm speaking with Kristina Grigaityte. Kristina is a PhD student at the Cold Spring Harbor Laboratory, and we will be talking about T cells and power loss. Kristina, welcome to the podcast.
Thank you for having me.
Kristina, tell us about yourself and about your background.
Okay. So I did my bachelor's at University of Edinburgh in Scotland, and that was in neuroscience. And I also did a year abroad at UCLA, and I worked in a couple of labs at UCLA and University of Edinburgh. These were neuroscience labs, so no coding, no nothing, just experimental neuroscience. And then I applied to a graduate program at Cold Spring Harbor Laboratories. They have their own graduate school there called Watson School of Biological Sciences, and got in and started my PhD there. The program that I'm in is quite unique in the sense that it's not specific to any given field. It's just a PhD in biological sciences. Usually it's already limited to either neuroscience or immunology. So the reason I'm mentioning it is because I was able to explore any topic in biology I wanted that was present at Cold Spring Harbor. And while my plan was to do neuroscience, I decided to do one laboratory rotation in a computational lab. I had no coding background, no programming experience. So I just decided to do it to pick up some coding skills and then continue on with my neuroscience career. It so happened that I ended up joining that lab, and my field changed from neuroscience to computational immunology. And the reason for that was just simply because I enjoy programming a lot and found the work interesting. And now I'm a fourth-year PhD student in a computational lab over there.
Pretty cool. So you just decided to try out programming and picked up immunology along the way.
Yeah, so it so happened that the project I ended up doing for my PhD was an immunology project. It was just what the lab was at the time.
And so you did a study. I'm not sure whether that's the same study as the project you were referring to, but you did this study of a big sequencing project of lymphocytes. And can you talk about the study and what motivated it?
Yes, so that was part of the project. That's actually the huge part of the project. And my lab, I'm in Mickey Atwell's lab at Cold Spring Harbor, and he had a collaboration with a company called Juno Therapeutics. They are now bought out by some other company. I'm not sure which one, but they had this new technology in their company where they were able to encapsulate a lot of cells in a high throughput manner in these emulsion droplets and barcode them and do single cell sequencing on them. So they were interested in doing these immune cells, T cells, and B cells. And so they used the technology to perform this high throughput sequencing on a huge number of T cells and B cells. And we were interested in T cells. So because of this collaboration, we ended up receiving that huge data. And this was the biggest throughput at the time, and I think still is today, although I'm not entirely sure. But as far as I'm aware, this one is the biggest one. And so I was very fortunate to get my hands on this data. The reason this data was so special is because of the way the T cell receptor is made. So the T cell receptor has two chains, alpha and beta chain. And previously, most of the studies only did bulk sequencing rather than single cell. So because they did bulk sequencing, they couldn't recover the paired information. So they had their beta chains, but they didn't know which alpha chain paired with which beta chain. So in the end, they only had half of the receptor. But because the beta chain is considered more complex than the alpha chain just because of the number of combinations that it could have. Most of the scientists would only look at the beta chain and assume that that's a correct way of looking at the T cell repertoire. So what's special about our data is it's in such a high throughput. We have around 200,000 alpha beta pairs that already post filtering. So those are high quality sequences. And the fact that we had the pairing information. So we have both alpha and beta chains, and we knew which alpha and which beta paired together. So because we had this opportunity to address the question whether the beta chain is an accurate way of looking at the T cell repertoire, we performed a bunch of analysis on this data. And ultimately, we show that while it's not too far off looking at the beta chain alone, rather than the paired T cells, we find that in some cases, most of the cases, independent of which question you're asking, it's actually much more accurate to look at the paired information.
Could you walk me through the technical details of the analysis? So from this company, you get a big, I'm assuming, FASTQ file or something like that.
Yes.
So basically all the reads that you have. And how do you proceed from there? So I assume the RNA that was sequenced wasn't pre-selected, right? So you have to extract these alpha and beta sequences from all the mRNAs out there. How do you do that?
So actually, I forgot to mention this, but this is targeted sequencing. So we did not have to extract it. It was already targeted to these alpha and beta chains of the T cell receptor. So my FASTQ file already contained these T cell receptor reads, either alpha or beta, and the reads were already collapsed by the unique molecular identifier. So what my data looked like is I pretty much had a sequence. It could have been alpha or beta. And I have a unique molecular identifier associated with it, and also a droplet barcode. The droplet barcode is when you encapsulate any given cell into a emulsion droplet. Each of the droplets have their own barcode. So we use that barcode to later identify which alpha and which beta came from that same droplet. So that's also included in the FASTQ file.
And how long are these chains, or the RNAs coding these chains? Are they short enough to be covered by a single sequencing read?
So I think, if I remember correctly, this was paired and sequencing. So there were two primers, one on the one end of the, let's say, beta chain, and the other one on the other end, and then they end up connecting in the middle. So, and also the technology used for this was MySeq. So they tend to have this a capacity for doing longer reads. So that's how it worked. So they ended up engineering the two sides together in the middle, and the output, I didn't have to do this. The output was already provided to me. Mm-hmm. So that's how it worked.
So basically you have, did you say 200,000 different cells, and you have basically two transcripts from each cell? Is that how it works? One for the alpha, yes, and one for the beta, yes, chain?
So originally my FASTQ files contain many more reads, many more transcript reads than that. But sometimes it happens that for a given cell, only a beta is sequenced or only an alpha is sequenced in a high quality manner. So we end up dropping those cells and only keeping the ones that we have both alpha and beta information for. So that's the 200,000 is already the both chains of it. Yeah. But originally we had many more reads than that.
And within a single cell, did you ever observe any diversity in those transcripts, or are they always completely identical?
Oh, they're very heterogeneous. So the way the T cell receptor is made, it's made in the thymus. And we have a huge diverse repertoire in our bodies just simply because there's so many bacteria and viruses that we need to fight against. And so we need a lot and lots of different T cell receptors in order to do so. So the way, it's actually very interesting that if we were to have one sequence for each T cell receptor, we would pretty much take up all of our DNA. So that would not be feasible. So the way it works is that there are a number of genes, and the different combinations of genes make up a T cell receptor. So for example, for the beta chain, there are genes called V for variable, D for diversity, and J for junction. And so there is I think there is around 50 V genes for beta chain, and then 13 J genes and 2 D genes, or along those numbers. Maybe now there's more. But the way it works is that for each beta chain, whenever the T cell is produced, one V gene is selected, supposedly randomly. Then the D gene is selected and the J gene is selected, again randomly. And then in the junctions, for example, where the V gene connects to D gene, there is also random repair going on in that junction site, plus some insertions and deletions. So the total estimate theoretical diversity, given these different constraints and insertion deletion rate and number of genes, for both alpha and beta combinations is about 10 to the 15. So that's how many unique T cell receptors we could have in our body. In our body, we only have reportedly from 10 to the 11 to 10 to the 12 T cell receptors, well, T cells with their receptors. So, but we can have any of the sequences from that 10 to the 15 in our bodies. So whenever I look at any given cell, it's very varied which T cell receptor it will have. And some T cell receptors I only see one time, and there are some T cell receptors that are very expanded, proliferated. Maybe the person encountered some disease just recently, maybe had a flu, and then I would observe a certain T cell receptor multiple times in a given sample.
Right. So that's when you look across all the cells that you have. But if you look at a single given cell and you look at the multiple mRNA transcript from that cell, are they all identical, or are there sometimes small differences between them?
So the way I get my data is I already have my transcripts collapsed. So I really only see one transcript, one alpha and one beta. So I can't really see if there's any difference between the transcripts in the cell. And even if I were able to see that and I saw differences, I wouldn't know if it was because maybe sequencing error or if it was any like a biological thing. So
one thing I thought about is that because we have 2 chromosomes, right? 2 of each homologous chromosomes, then we essentially have 2 copies, one of which is supposed to be silenced. But yes. Maybe if you sequence enough, you'll maybe see tiny glimpses of that supposedly inactive copy.
Yes. So I can answer this question, is that when the T cell is developed, so there is these V, D, and J gene recombinations happening during the production of the T cell. First, there is a beta chain rearrangement. And so there's only one allele that's undergoing this beta chain rearrangement. And once that's done, if the beta chain is productive, so it can produce a productive T cell receptor, there's no, shifts or anything like that, frame shifts, then the other allele for the beta chain is silenced. So it doesn't undergo the beta chain rearrangement anymore, as far as we know. For the alpha chain, once the beta chain is produced, then there is an alpha chain rearrangement. And for the alpha chain, it's reported that both alleles actually recombine. So you end up getting both alpha chains recombined, but then later there is some selection processes. So even if the full productive T cell receptor is made, it might still not pass a selection process. It could maybe not have very high affinity to anything, so that would be useless. Or it could it might have too big of an affinity to our own cells that are not a foreign cell. And so in that sense, in that case, we could result in some severe autoimmune diseases. So our T cells would attack our own healthy cells. So after passing those selections, most of the time, only one alpha and one beta chain receptor is made. So the other alpha chain that's also recombined doesn't pass the selection, so it's not there. It is also reported that in around 30% of the cases, 20 to 30% of the cases, depending on where you look, a given T cell will have two alpha chains successfully recombine and pass the selection. But for our case, because when you encapsulate T cells into emulsion droplets, we do it at a Poisson process to ensure that there is only one T cell per droplet every time. But Poisson process is not entirely without flaws in that sense that sometimes, and in our case, around 10% of the time, we will see a droplet that will have two T cells incorporated in it. So because let's say we see in a T cell two alpha chains rather than one with the same droplet barcode. So that could be one of the two scenarios. One is that we had two T cells incorporated into that droplet, and then one alpha chain came from one T cell, another alpha chain came from another T cell. Or another scenario is that there are actually two recombined alpha chains in a single T cell. And that could be because both chains passed the selection, and so the T cell potentially has two receptors. However, because we couldn't differentiate between these two scenarios, we ended up excluding those T cells from our data set. So we did see quite a number of the T cells that had both two alpha chains or even two beta chains, which shouldn't happen, but that could have been because we had two T cells in a droplet. And so for our purposes, we ended up excluding all of those T cells that had more than one alpha or more than one beta chain inside them.
Right. So you get this data set of various alpha and beta sequences for each cell. And then did you sort of know in advance what you were going to do with them? Or did you look at the data and thought, well, what sort of interesting insights can I draw from this?
So when I joined the lab, there was already another person working on this. So my colleague, Jason Carter, he is an MD PhD student at Stony Brook. And so he started looking at the data first. And what he saw is that the alpha beta pairs follow this distribution called the power law. And that already was reported previously in a single chain, but he ended up also finding that with the pair chains. So when I joined the lab, we already knew that the pair chains also follow this power law. And after he went back to medical school and I joined the lab, I ended up picking up the project. So I already had some pre-existing knowledge about the data, which made things a bit easier for me. And then after that, I just kind of went along with what I found and what I found interesting. And all of the rest of the analysis that I did just kind of came out as I went with advice from my PhD mentor and other people along the way.
Mm-hmm. So what does it mean to say that these sequences follow the power law?
Okay, so the power laws are a very interesting thing in that sense that they're everywhere. I never knew about it before I started working on this, and now that I know a lot about them, I pretty much see them everywhere now. So what a power law is, the way I like to explain it, even in my scientific presentations, I give an example of a Shakespeare play. So if you take a Shakespeare play, let's say take Hamlet, and you count how many times each word is used in that play. And then you take the most used word and then plot it on, you just plot from you rank them from the most used word to the least used word. And if you plot that, each word on the x-axis, you plot the rank of the word. So number 1 meaning the most common word and the last one meaning the least common word. And on the y-axis, you plot how many times you see that word. And if you make the scale log-log scale, so both x and y-axis are on a log scale, you will observe a very nice straight line. So that means that it's a power law. So the power law is pretty much an equation of y equals x to the power of gamma, right? And that's a power law. And so that's a very completely unrelated not related to biology example. And this kind of distribution we observe in many places. For example, city sizes follow the power law. City population sizes follow the power law. So my T-cell clone size distribution follows the power law. So if I look at my T-cell receptors that I have in my sample and count how many times that each of that receptor I see, so there is going to be a lot of receptors that I only see once, and there's going to be a few receptors that I see many, many times. So if I plot that distribution the way I explained the Shakespeare example, I would also get that straight line on a log-log scale.
Yeah, I just wanted to clarify one small thing. So I think there are two alternative ways to describe, or probably there are more, to describe a power law. And what you were talking about originally, the relationship between the rank of the word and its frequency, right? That's, I think it's called Ziff law.
Yes, that's correct.
And there the relationship is simpler. It's, I think it's like something like the frequency is inversely proportional to the rank. Mm-hmm. But if you turn that, but that's if you can calculate the rank. Mm-hmm. But an alternative way to approach it is, instead of calculating ranks, you just consider simply the distribution of the frequencies. Yes. And you say the higher the frequencies, the smaller the probability of observing it, and the relationship between the frequency and the probability of observing that frequency is the power law, meaning that the probability is proportional to the frequency to the power of gamma.
Yes. So
just not to confuse those two things, because they are related. I think they're describing the same underlying probability distribution, but one way to describe it is in terms of the ranks, and the other is in terms of the distribution of the, I guess, cluster sizes. So
the way I like to say it is that, yes, you're right. The rank way of doing things is called Ziff's law. And then what we do with T cells, instead of just plotting the counts of the word on the y-axis, we actually count how many times we see the count. So I like it to call it counts of counts, kind of, or frequency of counts. And then the count goes down to the x-axis, and then the frequency of that count goes to the y-axis. So for our T-cell example, we're doing the so-called counts of counts. And that still remains a power law. And what's actually interesting is that if I take some, very big sample, let's say I have a sample of 1 million things, can be anything. Could be T cells, could be, for example, how many times a given tweet was liked. So likes of tweets also follow power law. So let's say if I have a huge sample size and then I take the counts of counts, I get the power law. But then if I count the counts of counts, I still get a power law, and I can do it many, many times. And no matter how many times I count the counts of the counts, I remain in the power law. So I thought that was very interesting, and I kind of accidentally discovered that while I was playing with some data.
That's interesting. Mm-hmm. And so another thing, I guess, to note about the power law, because it may seem like very abstract. So you have some kind of formula with a power in it. So the significance, I guess, of this formula is if you compare it to other distributions, so things like the normal distribution or the exponential distribution, they decay very quickly because they're an exponential with a negative power. So this is why the range of values that you can observe is pretty narrow. But the function x to the power of something, to the power of gamma, it grows. If gamma is positive, it grows slower than an exponential. And if gamma is negative, it falls slower than the exponential. And this is why for very large values, you still have relatively high probability of observing it. And this is why the range of the power law distribution is typically much, much larger than for other distributions. That's why sometimes called a heavy tail distribution, because it decays relatively slowly compared to the various exponential distributions.
Yeah, that applies if you have a low exponent. So the lower the exponent, the heavier the tail. If you have exponent that's pretty high, let's say exponent of 4, you'll actually have a very low probability of observing anything above 10 on an x-axis. That's something I also discovered by just playing around. I can tell you another interesting thing.
Yeah, go ahead.
So another thing that power law resembles is something called a Pareto distribution. So Pareto distribution is pretty much like a power law, and it kind of says that 80% of the problems are caused by 20% of the causes. So for example, 20% of the richest people owe 80% of the earth wealth, or 20% of bugs cause 80% of software problems. So that's another thing. So in a way, with the T-cell example, it means that about 80% of our T-cell repertoire consists of only 20% of some T-cell receptors in terms of diversity. So I thought that was also kind of interesting and something you can infer from the power law as well.
Right. So what is the biological basis of that sort of inequality? How come that the, let's say 20%, but I guess the specific figure of 20% and 80% would depend on the power of the power law?
Yes.
But like, how come that the minority of T-cell receptors dominate the whole population?
So that's something that nobody really knows. So for whatever power law you look at, whether it's in biology, whether it's in economics or, geography, nobody knows why they happen. So we don't know that in case of T cells either. There are some explanations that could be possible, but these are the kind of explanations that, they could be right because they make sense. There is no really theoretical proof for them. In terms of T cells, I guess biologically, in a way, it makes sense to me that a huge fraction of the population is only a few T cells, or 20% of the T cells, is because probably of the history of it must have to do something with evolution, probably, is that certain pathogens are very common that humans get exposed to, while others are not. And so maybe humans built up this kind of repertoire. There's absolutely no biological explanation that's proven in any way. It's just my speculations right now.
Okay, I kind of expected the answer to be it's just the clonal expansion, but maybe that's more complicated than that.
It's probably more complicated than that. In terms of T cells, there's many different types of T cells as well. So, for example, there is a CD4 and CD8 T cell. CD4 T cell is another way called a helper T cell, and a CD8 T cell is called a killer T cell. So, usually, when you have an infection, the helper T cell the helper T cell is the first one to react, and it recruits the killer T cell, and then the killer T cell does the fighting. And so the killer T cells are the ones that have a higher rate of proliferation. And so they're probably more likely to be at the tail or will follow a more heavily tail distribution with a lower exponent. So this could be one explanation that there's some, T cells that proliferate various rates given an infection, and then some T cells have a less proliferative rate, and somehow it comes together in a way that it follows a power law. Another type of T cells are memory T cells and naive T cells. So naive T cells are the ones that never encountered a pathogen before. So they usually tend to be in very low clone sizes, just simply because they didn't have an opportunity or reason to proliferate. And then memory T cells are the ones that have encountered a pathogen before, and then our body wants to remember that T cell just in case we have the same encounter again. And so that T cell tends to stay at a slightly higher clone size, just in case it will need to perform again. But another interesting thing about the T cell case is that as I mentioned earlier, the power law on a log-log scale appears as a straight line, and that's how you can suspect that it's a power law. In the T cell case, the power law actually deviates slightly from the straight line on a log scale. And at first we thought maybe it's just noise. But then, again, I was playing with the data, playing with some simulations one day, and then I decided, I don't remember why I decided to do this. I decided to just simulate two power laws of different exponents and combine them together and see what I get. Do I still get a power law, or do I get something else? And I still get a power law, what I think is a power law, but there's a slight deviation from that straight line. And then I noticed that it's a very similar deviation from the straight line as my T cell case, my T cell power law. So then I thought, wow, maybe the T cell receptors follow a mixture of power laws. Maybe there's more than one power law in there. And so that kind of led us to explore mixtures of power laws. And actually, when we thought about it more deeply, is that the different types of T cells present in our bodies could potentially explain why there is a mixture of power law, because there could be different generative processes. So for some T cells, they're more likely to proliferate, while other T cells tend to stay in low sizes, and that could lead to existence of two power laws. And since we didn't differentiate between the types of T cells, we ended up looking at the whole thing in one go. And so we didn't separate them by type, so we couldn't separate the power laws. But we don't know whether what I'm saying now is true, just because we don't have the information for cell types. But that's to be determined in the future. But that was something completely novel, that we saw that there's a distribution out there that is a power law, but in fact, it's not a single power law. It's more than one power law. Interesting.
And I guess the reason why you couldn't check within the subpopulations of T cells, like separately CD4 T cells, is that you did this targeted RNA sequencing. So if you did the whole RNA sequencing, you could presumably cluster them by type, right? And look within the cluster.
Well, so actually for CD4 and CD8, we did have that information.
Okay.
So the way it worked in sequencing is that they use these barcoded antibodies. So CD4 and CD8, these are surface proteins. So you can get an antibody to bind to, let's say, a CD8 surface protein and get encapsulated along with a cell. And then you can have a barcoded antibody attached to the CD8 protein and then sequence that barcode of the antibody and then determine whether the T cell is a CD8 T cell or a CD4 T cell. So we actually had that information in our data. Not all of the T cells successfully carried that along with them during the sequencing. So when we divided the T cells by CD4 and CD8, the sample size decreased quite a bit. So we no longer had 200,000, but we still managed to look at individual power loss for CD4 population and a CD8 population. And we did see that CD8 population tend to have a lower exponent compared to the CD4 T cell population, which again makes sense because CD8 T cells tend to proliferate more. So in that sense, this suggests that maybe one of the reasons for the mixture of power loss is that CD8 T cells proliferate more. But within those two populations, both CD4 and CD8, the best model to fit is also a 2 power law model. So we calculated, the best model was calculated using this thing called Bayesian information criteria. And so we found that the better model is the 2 power law model. And another thing, so that's, there's a package that I created to determine this. We could also determine what weight each power law takes up. So when we observe 2 power laws in the full population, we see that about, I think 10% of the distribution is a power law with a lower exponent. And then the remaining 90% of the distribution is a power law with a higher exponent. For CD4 T cell population, only 1% of the distribution is the lower power law exponent, so the heavier tail one. And then the rest, 99% of the population is the power law of a higher exponent. For the CD8, we observe a higher weight of the lower power law exponent. So again, we have 10% of one power law and 90% of the other power law. And so this suggests that there's also the difference in how much each of the power law take up in CD4 versus CD8. And both CD4 and CD8 have memory and naive cells. But, and I'm not too familiar yet on what's exactly the percentage of memory and naive within a given population. But CD8 T cells, I would think, would have potentially a higher number of memory T cells than CD4. But I'm actually not sure about that now. So there's differences in what's the combination of different mixtures in CD4 and CD8. But since we don't have memory and naive information, we can't really continue dissecting the population even further.
But again, if you had a full RNA, full transcriptome profile of the cells, couldn't you then try to separate the memory and naive cells based on the gene expression profile?
Yes. So this could be a strategy to use just to do the whole transcriptome and then extract T cell receptors, memory or naive transcripts or whatnot. The problem with that is that we would probably need much more coverage to do this because we want to recover our T cell receptors as accurately as we can because that's the best way to determine clonality of the repertoire if we only have sequences that are very, very accurately sequenced. And the only way we can have that if we have lots of reads for a given T cell receptor chain. If we do a whole transcriptome, then all those reads would distribute among many different transcripts, a lot of transcripts that we probably don't care about, for at least for this given project. So that's why this scenario or this protocol would not have worked for us because we could have potentially missed a lot of T cell receptors. And this would have been probably much more expensive since we would have needed a much bigger coverage for the sequencing. So
I see.
Yeah. But there's other strategies to get the, CD4 and CD8 information the way we did in this project, or for memory naive. Actually, in the next project that I'm doing, we are extracting memory naive information, again, using this antibody technique where we attach the antibody to a surface marker and then see which surface marker attached to, whether it's memory or naive or CD4 or CD8. So there's other ways to extract this information without sacrificing the T cell receptor quality.
And since you have the information on the alpha and beta chains of the T cell receptor, then I guess you decided to investigate the relationship between the two. And you already mentioned that the consensus was to pay attention to only one of them.
Mm-hmm.
How exactly did you find the link between the two?
So the main idea for the paper was really to compare the paired T cell receptors to their individual chains. So the way I did this was I had my 200,000 alpha beta pairs, and then I took my data set and also split it by chain. So I don't have a change in sample size. Sample sizes stays the same, and I just have 3 pretty much data sets: one for the pairs, and then one for the alpha, and one for the beta. And so then I wanted to perform same analysis for each data set that I now have, each of the 3 data sets, and then compare what kind of answers I get. So usually people only look at the beta chain and assume whatever conclusions they make, these are correct conclusions. So this way of doing things, the way I did it, was to just directly compare what answer we get from just the beta chain or just the alpha chain to the answer we get from the paired. And the first thing I did is I kept exploring the power laws. So it was very simple. Idea is just to fit the power law distributions of paired and then compare them to the fits of only alpha chain and only beta chain. So this idea that if the pairing of the chains is completely independent and looking just at the beta chain is an accurate way of doing things, then those power laws should pretty much be the same. Maybe a little bit of noise, but they should mainly have the same exponent and really no significant differences. But that's not really what I saw. In fact, when I looked at the pairs, as I said before, I observe a double power law, so that a model of 2 power laws is a better model than a single power law. For the beta and alpha chains, that was not the case. The better model was a single power law model. So that's a pretty significant difference. And that was pretty much the first analysis I did that really showed that the paired data set is a much better way of looking at T cells. And that makes sense because when you only have a beta chain, you pretty much have half of the receptor. And you assume that the other half is not important. And this pretty much suggests that the other half is important. Another thing that we did was to look at the number of alphas one beta chain pairs with. So we take a given beta chain, one beta chain, and then go through the data set and see how many times that beta chain pairs with some other alpha chain in another T cell receptor. And then if the pairing is independent, the most likely outcome of this should be that pretty much all of the beta chains only pair with one alpha chain.
So sorry, I'm not sure I follow. So if pairing is truly independent, then every alpha chain and every beta chain should have like an equal probability to pair with each other, right? That's what independence means?
Yes. But because there's so many of potential combinations for alpha or beta, it's very unlikely that the same beta is going to be generated in 2 T cells more than once or twice.
So you're saying if we are observing the same beta in 2 different cells, then it cannot be by chance. They have to be descendants of the same clonal expansion.
So it can't be So the only way it can be a descendant of the same clonal expansion if it has identical alpha and beta chain.
Right.
And here I'm talking about the beta chains that are a same beta chain in two different cells, and they pair with different alphas, or other way around.
Right.
A single alpha pairing with two different betas in different cells.
Yeah. So therefore they're not independent, right? Yeah. Is that what we're saying?
That's what we think it is, that they're not entirely independent, and the independence could come from many different ways. Maybe, some alpha beta pairs are not likely to be a good T cell receptor. So if the T cell production is unsuccessful, then our bodies just keep producing T cells that are successful. And maybe, this kind of selection also lowers the estimated diversity, or estimated potential for diversity rather, of what kind of T cells could be generated, because the production of T cells happens before they get exposed to any pathogen. So they don't know what they will need to fight against. So the body just pretty much generates a ton of different T cells, right? Presumably randomly. And we think that this generation of T cells, including the pairing of alpha and beta chains, may not be as random as thought before. We don't have direct, experimental proof for it or anything like that, but our analysis kind of suggests that this could be the case.
And so there is a dependency between the alpha and the beta chains, but so they're neither completely independent nor one of them determines the other one, right? Somewhere in between there's like sort of correlation.
So we don't know. So as of now, it is widely believed that the pairing is completely independent. There's different compartments in the thymus where the chain recombination happens. So the beta chain recombination happens in a compartment different from the one where the alpha chain recombination happens. So as far as current knowledge tells us is that the recombination of the chains should be independent. But I have reason to believe, based on my experience with this data and, pretty much working every day on it, that I don't think this is as simple as independent. So there has to be something more than that, because if it's only independent, then we wouldn't be seeing what I have seen in this data.
Okay, so basically you have now a result that more or less completely overthrows what previously was believed, and also we have no idea why it happened. So it sounds like a great starting ground for a very successful research career.
Hopefully, I don't know. We'll see.
Like a lot of questions to answer here.
Yeah, it's a bit of a controversial idea, because so much work has been built upon these assumptions, that the pairing is independent, and so much work has been done only on the beta chain, including work in medical fields, for example, for, cancer immunotherapy, a very hot topic right now. Cancer immunotherapy just got a Nobel Prize this year. And a lot of the work that's done, in clinical trials is only looking at the beta chain. And I guess the message that I'm trying to tell here is that maybe we should, be more responsible in the sense of making those assumptions. And I actually show that looking at the pairs is a much more accurate way of, doing this kind of science. And maybe, there should be a move from this established bulk beta chain sequencing to single cell sequencing, especially given that recent technologies has been growing very rapidly.
I guess one thing I don't quite understand here is that if people believe that a pairing of an alpha and beta chains is independent, so meaning any alpha chain could be paired with any beta chain, then I don't see how it follows that, like, how they justify of looking only at the beta chain, right? If you believe that they're independent, then the pairing of alpha and beta carries the added information of alpha plus the information of beta, right?
Yes.
And so that would be, like, if people believe that, that would be only an argument for looking at both of them.
Yes. So I think while this T-cell repertoire field was developing, it was very hard and very expensive to do single-cell sequencing. It still is quite expensive. So I think scientists didn't really have a choice. That's the best that could have been done. And the reason, they do beta chain is because beta chain has a selection from 3 types of genes, while alpha chain only has a selection from 2 types of genes. So beta chain has V, D, and J genes. Alpha only has V and J. And also, it's previously reported that the beta chain is the chain that has these insertions and deletions in the junction sites, while alpha is believed not to have those extensive insertions and deletions, and that makes the beta chain much more variable than the alpha chain. And because it's, so many different combinations that can be made given all these constraints, it's supposed to be a very low probability of observing, one alpha chain pairing with different beta chains in different cells and other way around. And so that led to this assumption. But the assumption is purely based on the belief that there is this independence, right? And that a given beta chain, it's a very low probability that the same beta chain will be generated in 2 different T cells. And we see that happens a lot. Another interesting result that we saw was T cells that were shared among individuals. So, for example, assuming that there is, 10 to the 15 possible alpha beta pairings, and humans only have 10 to the 11 T cells in the human body, it's very unlikely that 2 unrelated individuals will have the same T cell, just because, you have a probability of generating any given T cell is, 1 over 10 to the 15, and you only have 10 to the 11 T cells in the human body. So it's, very close to 0 probability that the same T cell will be generated twice in an unrelated individual, or even within the same individual. And we observe paired alpha betas among different individuals, different unrelated individuals. And if this independence assumption, is correct, and we shouldn't see that. Even given the number of possible combinations, we shouldn't see that. The probability of observing these 2 sequences is so low and we see way more than the probability should suggest. Mm-hmm. So that's another piece of result that suggests that there has to be something more in the way these T cells are generated and the way the alpha beta are paired together.
Right. I guess this could also be explained if the combinations of different segments is sort of non-uniform. It's random, but some combinations are more likely than the others, and there may be a few combinations that are like super popular or super frequent.
Yeah, so if you only look at the genes, then you will see that some genes are more likely to get selected than others. And that could be, for example, maybe, when the T cell receptor is folded into the protein, maybe, certain gene combinations don't really fold well, into a stable protein. So, those combinations are not favored. However, when we consider, a T cell receptor, we also consider the junction sites. So actually the junction sites are the most important sides of the T cell, in that sense that that's the site that recognizes the pathogen. So that's not only the genes, that's also insertions and deletions. That's the region called a complementarity determining region, or a CD3 region. And so we take that full CD3 region to determine a T cell receptor. So, and that's another thing, is that it's not only gene selection, it's also the randomness of insertions and deletions. And the insertions and deletions are believed to be completely random. And if that's the case, if those insertions and deletions are indeed random, then again, we shouldn't see shared T cell receptors among unrelated individuals. And the fact that we do see that also suggests that maybe those insertions and deletions are not as random, or, something is going on there that causes certain combinations happen more frequently than others.
Yeah. And speaking of alpha and beta chains, he also had an interesting observation about how the pairing of alpha and beta affects the specificity of the receptor to like different epitopes.
Oh, yes. So that's kind of been known for a while in mice. People have done, mutation experiments where they took a T cell receptor and changed one amino acid on the alpha chain and observed a completely, changed specificity to a given epitope. What I did with my data is there is this database that exists right now online. It's a vdjdb.net or something like that. It's a T cell receptor database. And it has some paired T cells from other single cell experiments and a lot of single chain alpha or beta sequences there that you can look up. And also, all of these sequences have a specificity information. So maybe they were determined using these tetramer assays where they, put in a given epitope and then pooled all the T cells that reacted to it and sequenced them. And this way they knew which pathogen is that T cell specific to. And so what I did is I took my data and looked into that database to see if there's any T cells in there that I also have in my data set. And I found quite a few of them. So I looked at both at the paired level and also the single chain level. So again, I did the same thing. I took all of my paired T cell receptors and then looked how many of the paired T cell receptors I see in the database. So I found five that are shared between the database and mine. That's again another thing. I shouldn't have seen that, given that the recombination is completely random, right? So that's kind of a reproducibility thing. Like I reproduced that with a completely different data. So that was another thing that was really cool. But what I also did is, again, I split my data set by chain, and I also split all the paired sequences that I saw in the database also by chain. And then I saw what I would find. Do I find any shared sequences only at the alpha and the beta chain? So what I found was very interesting. I actually found much more only single alpha or single beta that are shared between my dataset and the database. So then I looked at the epitope species that those T cells are specific to. So if I take one individual, and there was one individual that shared T cells at the paired alpha and beta chain level with a database, and those T cells were specific to influenza A, which is just a general flu virus, and EBV. These viruses are very common viruses that a lot of people have T cells reactive to those viruses. But then if I look at single chain level, either alpha or beta chain, all of a sudden I see that the same person that had the EBV and influenza A now also has HIV, yellow fever, cancer, and a bunch of other viruses. And, if we were to use our T cell receptor sequencing for, maybe diagnosing, just hypothetically speaking, it's not done right now or anything, but if we were to do that, just to use half of the receptor, so only the beta chain, to determine what kind of virus that person is exposed to, So if we only looked at the beta chain, we would conclude that the person has all of these horrible diseases, and that would be quite unfortunate for that person. But if we look at both pairs, if we look at both alpha and beta chain together, then we only see influenza A and EBV, which makes much more sense and is actually a common thing to see in the individual. So that was a pretty striking result when comparing alpha and beta pairs to just a single chain.
Mm-hmm. But that's only assuming that your cells are memory cells, right? Because if those are naive cells, you kind of make the assumption that the person actually encountered those viruses.
Yeah, that's actually a good question. I never thought about that. I'm not sure how to answer it fully. I guess, we would have to know the memory naive information. But I think in our case, when you sample a T cell, so we sample T cells from a peripheral blood. So we're probably much more likely to sample a memory T cell over a naive T cell.
Yeah. Because
memory T cells are at higher clone sizes. So it's a much higher chance that the T cells I looked at were in fact memory. But the only way we can answer this question is to actually know the cell type.
Yeah, that makes sense. Okay, so one conclusion you reached was that the alpha and beta chains are not independent. Any other results from that paper that you want to talk about?
Not from the paper. I have a really cool but very anecdotal example that we didn't include in the paper just because it was anecdotal. But I always share it in my talks. So one of the things that I noticed, so originally in our data set, we had 6 healthy people. And healthy here means that they don't have cancer and they don't have autoimmune disease. So they shouldn't have these, random expansions related to cancer or autoimmune disease. And in our final paper that we put on the bioarchive there were only 5 individuals. The 6th one wasn't there. And the reason for that was because when we looked at the power law distributions of each of the individuals separately, we found something very strange with the power law of that 6th individual. The strange thing was that individual had huge expansions. So much bigger expansions than any other individual. And the power law was looking kind of weird in that there were very few data points at the head of the power law. So, clone sizes of 1, 2, or 3, they were there, but then there was a huge gap, and then there were only a few data points of these very high clone sizes. And so we found that there has to be something going on with that person. And we actually ended up reaching out back to the company that we were collaborating with and who did all the, blood collection and everything like that. And we asked whether they know anything about that individual and if there's anything, going on with that individual. And they told us that they found traces of hepatitis C in the blood. So we thought that was really cool, in that sense that we could see that maybe something is going on just simply looking at the power distribution of that individual. So then we just assumed that individual was not healthy. And so we ended up excluding it from our analysis. But I still share this in every talk that I do. And I show the power laws in every talk that I do, just because it's an interesting idea that those power laws could be used, to potentially infer something going on with a person's health.
Yeah. So in that case, was it the case that the power law was a bad fit for the data, or was it a good fit but with a different exponent?
So the fit, we didn't end up fitting. That was still back in the day when we weren't fitting power laws. We were just kind of observing them. Uh-huh. But the way the power law looked was that there were, 3 points at the head that were in a straight line. And then there was just a few points in the tail. So there were only very few, these clone sizes that we could plot to begin with, just because the sample was so dominated by these, huge expansions so it was kind of looking like a power law, from a clone size of 1 to a clone size of 3. But then after that, that was pretty much it. Then there was just these huge expansions. So we think that had that person be healthy, we probably would have seen a much nicer looking power law. But because there was something going on there was this huge deviation, or, the tail was so heavy that it completely dominated the power law.
Right. By the way, when you identify these clone sizes, how do you cluster together the sequences? Do you have some kind of threshold of similarity that you allow, or do you consider identical sequences only?
Yeah. So we look at these regions called CD3 regions, right? The ones that I just described earlier. And the reason is because they are the ones that, are complementary to the pathogen epitope sequences. And so I looked at those regions, and I allow for one mistake. So if there's only one nucleotide difference between the CD3 regions of 2 T cells, then I assume these T cells to be identical. The only exception for this was when I looked at the shared T cells among individuals. I wanted to reduce, error as much as possible. So I only considered identical, every single nucleotide is the same, in that case, just because, I didn't want to, other people come back to me and say, oh, that's because maybe, you were overcorrecting for sequencing error. So in that sense, that was, I looked at only identical ones, but for the power laws, and for estimating clone sizes, I allowed for one mistake, one difference.
And so for fitting power laws to the data, I think in this main paper, you use the maximum likelihood procedure, which is basically finding the exponent that maximizes the total likelihood, total probability. But you also wrote a Python package and released a preprint describing it, where you do Bayesian inference of power laws. And can you talk about that, and why were you not satisfied with maximum likelihood?
So the reason we used maximum likelihood is because that was, a quick way of kind of getting the exponent. However, when you use maximum likelihood, you only get one number. So let's say if I have my CD4 and CD8 distributions, I will get one number for CD4 and one number for CD8. And then what do I do with these two numbers, right? I can't really compare them. I don't know what's the certainty of these numbers. What's the confidence interval? And that was the flaw with maximum likelihood. With Bayesian inference, you actually sample the full posterior distribution. So not only you get, the maximum, you only get the spread. So you get, in our case, we were lucky to have a convex, a distribution in that sense that we got a nice one peak Gaussian. So not only we could, determine the mean of the Gaussian and get that number we would have gotten the same number with maximum likelihood, but we also have a confidence interval now. So that would allow us to actually compare the two distributions together rather than two numbers together. So that was the motivation for Bayesian inference. And this type of fitting of power laws didn't exist, or at least didn't publicly exist. And I thought, since power laws are everywhere, not just biology, and a lot of people are studying them in all types of fields, it could be a useful tool for other people to use and use Bayesian inference to fit those power laws without, having to write the full algorithm themselves. And yeah, that's why we did that as well.
And surprisingly, for some of the data sets, you found rather significant discrepancies between the maximum likelihood and the Bayesian mean, I think.
Mm-hmm.
Do you have any ideas why or any explanations?
So there's a few times, a few scenarios where fitting using Bayesian inference worked better than maximum likelihood. One was that when exponents were very, very low, close to 1, for some reason, the maximum likelihood would overestimate. So it would estimate a higher exponent than it actually is. And that's quite dangerous in that sense that a lot of power laws, most of the power laws actually, have an exponent close to 1. And another case was if you have a very low sample size. So Bayesian inference will still do a pretty good job when you have as low as, 10 data points, which I couldn't believe that was successful, but for some reason it was. And for maximum likelihood, with low sample size, it gets much more inaccurate if you have a high exponent. So depending on what kind of power law you have, sometimes using maximum likelihood would be sufficient and, faster, and that's fine. But there are some cases where you want to use Bayesian inference and, I don't know why. I think the main reason the Bayesian inference was superior to maximum likelihood because it provides that spread, that confidence interval. But I think doing maximum likelihood is, not completely inaccurate in most of the cases because a lot of the cases you'll have high sample size and, should be able to do well with maximum likelihood as well.
For small sample sizes, it sort of makes sense that the Bayesian inference would work more robustly because you have a prior. I think you give it the Jeffreys priors.
Mm-hmm.
And so you have this penalization that doesn't allow it to go completely insane. But in general, if you have a Bayesian point estimate, such as the posterior mean or posterior mode, and if you have a maximum likelihood value, so you said that the Bayesian values were more accurate, but how do you judge which one is more accurate when they diverge?
Well, when I did, validation of my algorithm, I do simulations. So I simulate a power law where I know the right answer, right? So that's how I know.
And so even when you simulate, so initially I thought that when you got different estimates for two methods, it was because maybe the power law wasn't a great fit. And so there was sort of ambiguity of how to interpret the data. But you're saying even if you simulate and you draw your data from a perfect power law distribution, you still get the discrepancies in the two methods?
Well, the differences between maximum likelihood and Bayesian are actually not that big, right? The only exception to that is when I look at the very, very small exponents that are very close to 1. And I actually don't really know why that happens with maximum likelihood for me. But with Bayesian, it doesn't happen. So that's why for me, it seems better in that sense.
Yeah, and it's interesting that you say the exponents are often close to 1 because those are very dangerous distributions, right? They don't have a mean value. So that means as you continue to draw from that distribution, your sample mean will become larger and larger in the limit. So like if you observe a human body, the counts of the cells will become sort of larger and larger as you continue observing it. If you didn't tell me that, I would imagine that that would be, you know, a bit maybe biologically or physically implausible, but I think that, yeah, that happens as well.
Oh, yeah. I think because there's always, you know, the tail is kind of like an outlier in a sense that there's, you know, very low frequency of these, you know, for example, in T-cell case, high clone sizes, right? It's, there's only once that you'll see, you know, a clone size of 1,000, for example. And many times you can see a clone size of 10 just because it's more likely to sample something 10 times than 1,000 times, right?
Right.
So, but because there's always, you know, some, like, for example, T cells that will be at such a high clone size that you will sample a lot of them, although rarely, that the power law is bound to have a tail, like a heavy tail. Mm-hmm. And the heavier the tail, the lower the exponent. And this kind of makes sense that since you would always have the tail, so your exponent would always be low and close to 1.
Yeah, and I guess, I don't know how relevant this is biologically, but when thinking about like extreme outliers, because these power laws, especially with exponent less than 2, they're bound to have huge outliers. And you could say that a cancer, like a lymphoma, would be one form of such an outlier.
Mm. So like, yeah, if somebody has, cancer, it would probably have, you know, a T cell clone size distribution even closer to power law, it would have a clone size distribution power law even closer to 1 exponent, right?
I was saying that you could consider that as something drawn from the same distribution, mm-hmm. but you happen to reach that very far end of the tail, so you get a very bad lottery ticket.
Yeah, I guess.
So I think we covered the T cell receptors and the power laws. And is there anything else you would like to talk about?
I think what is very interesting, you know, from my work, at least to me, was that I went from, you know, something like T cells and learning about power laws in a, you know, just in the T cell case. And now, I think I mentioned that at the beginning, I see them everywhere, right? So since then, I discovered more power laws that weren't seen before. So now we're trying to develop this single cell sequencing protocol, where we also use encapsulation, but we use this thing called multiplexing, where we, instead of using a Poisson process to encapsulate T cells inside the droplets, we barcode samples and then overload the emulsion. So this way you can have multiple T cells in a droplet, but because the different samples are barcoded, we can distinguish whether it's, you know, a single T cell in a droplet or multiple T cells in a droplet because of those barcodes. So this is under development right now. And we did a few pilot runs for this, right? And there were one pilot that wasn't very successful in the barcoding of the samples, in that we got a lot of random barcodes from somewhere else. So we were supposed to have, you know, 5 barcodes that we should have seen and sequenced, but we got, you know, millions of barcodes that were completely different from the ones that we were supposed to see. So what I did, I counted how many times I see each barcode and then plotted those frequencies of those counts, and I got a very nice looking power law. Yeah, so I see power laws everywhere now, and the most fascinating thing about them is that nobody knows why. Yeah. You know, we see them in T cell case, we see in a small sample. So maybe it's the way, you know, we analyze data. Maybe it's the way of, you know, humans looking at things rather than the actual phenomenon. And maybe that's just a simple explanation like that. So like in T cell case, we only sample, you know, a small fraction of the total T cells in the human body. And so because of, you know, this kind of sampling, a power law is just an artifact. But then there's examples where we do have the whole sample, the whole population, like, in languages. So if you take, every single word in the English language and count how many times the word is used in somewhere, let's say in this episode that we're doing right now, if we count each word, we have the full population, we have every single word, right? And we still see a power law. So it can be just a sampling artifact.
Well, I'm not sure I agree that that's the full population, because the episode itself is sort of a sample, right? We could have a different episode and, or use different set of words on a different day.
True. But I mean, you can also try and, take a full language. I guess you can't really determine how much time each word is used, all of the world.
It's still a sample, right?
Yeah. But when you take, a play and you just want to understand the words used in just the play, like a Hamlet play, you don't just sample a small fraction from that play. You use the whole play. So in that sense, it's the full population, because that's the play you're interested in. You have the full population of that play. And then another interesting thing is that, language is something that, was created by humans. So it's not, T cells, that's, nature, that's not really controlled by humans. And then there's language that's controlled by humans, or, city populations and stuff like that. And these two things, that one is controlled by humans, one is not controlled by humans, and it comes, together in the power law. I think it's pretty cool. And something to think about. And I think I read it somewhere that if we understand only one scenario of where the power law is coming from, like if I somehow succeed to understand why T cells follow the power law, then we can potentially understand why there's a power law in every single case. That's not my idea. I read that somewhere, but I thought that was really interesting. Maybe potentially understanding T cells could lead to understanding, language or something like that. So I thought that was really cool.
Yeah, it's interesting. I also thought about this, like if you contrast the power law with, let's say, a Gaussian distribution. So for a Gaussian, we know how it at least may arise if you sum up many independent observation, or you average them out, you get a Gaussian in the limit. So we sort of know how that can be generated. For the power law, right, we don't know anything similar. There is this scale-free argument. I can't say I find it too convincing, but the argument is that this is a spatial distribution that doesn't change if you rescale your variable.
Oh yeah, mathematically that's you can show that if you rescale, you still get the same answer.
But it's not clear why would it apply in practice.
Yeah, the why is not clear. Actually, there's another thing that I did that's, not published or anything, but, when I thought that, the power law, I still kind of think that the power law could be an artifact, just the way humans like to look at things. You do counts of counts. It's like a very special way of looking at things. And maybe it's just, that's just the artifact of the way we analyze data. And I thought, what if, since I'm sampling a small fraction of T cells, what if the actual distribution of T cells, if you look at the whole 10 to the 11 T cells, what if that's a Gaussian? And when you subsample, you get a, power law. So I tried simulating a Gaussian and then playing around with it and trying to get a power law out of it. And I almost succeeded. I got like a nice, straight line on a log-log scale at the head of the line, at the head of the distribution. And then I got like a very messy, like not power law-like tail. But I don't know. I thought you playing around a little bit more, maybe subsampling a bit more, you could turn a Gaussian into a power law.
I guess that means that we should be careful when we declare something to behave as a power law. We should have some kind of null hypothesis against which we test, or a set of hypotheses. So I saw this paper. It was about the power law, but the authors also gave some counter examples. So the distributions that have a very wide range So you might think they're power laws, but they're actually not. They resemble other distributions more. They don't quite follow the power law, but follow some other distributions. Although from a general description, like there's these huge outliers, there are heavy tails, you might think it's a power law or might want to apply a power law to them.
I guess, like sometimes you could confuse maybe an exponential with a power law, given, what parameters the exponential has. And maybe, a combination of many, many, many exponentials could resemble a power law. That's something that I guess I would have to, simulate a bunch of exponentials and see if I can get something close to a power law. But that's something that could definitely happen, Playing around with other distributions could, lead to a power law. And yeah, so you have to be careful. I think when I was first learning about the power law, I was just, Googling Zipf's law and power laws. And I think I came across this one entry, some web page with a title, so you think you have a power law? And like, do you really have a power law, or do you just think you have a power law? So yeah, you have to be careful. And there's definitely tests, like statistical tests that people do to determine whether, it's really, following that power law model, or if it's deviating and how much is deviating from it and stuff like that. Yeah.
Cool, Kristina. It was a very interesting conversation. Thank you for taking the time.
Oh, yeah, no problem. I was happy to do this.